Ferns reproduce through spores, and clusters of spores in sporangia can be seen on ferns in various places, sometimes on the backs of leaves, sometimes, in the case of interrupted fern, in the middle of the leaf. So what you see are not dead leaflets, but rather the spore-producing ones. Or sometimes there are separate leaves which only produce sporangia, so they are not green. So these are not dead leaves. Instead, these are where the spores are being produced. What do spores germinate into? Well, not into the fern that you are accustomed to seeing. Instead, ferns have two parts of their life cycle. They have an alternation of generations. And what develops from the spore is what is known as a prothallus or prothallium. It is a small heart-shaped structure, only 2 to 5 millimeters in size. So in this image, you see a number of prothallia next to a dime for size comparison. These are typically green and capable of photosynthesis, but they can be colorless and decompose some of the surrounding rotting vegetation. They typically only live a few weeks before they develop into the type of fern that you are accustomed to seeing, what's known as the sporophyte. And the sporophyte will briefly depend on this prothallus for its nourishment. This prothallus represents the gametophyte stage of the fern, where the cells are haploid, and this might take a little explaining. In humans and in other animals, we think of haploid cells only in terms of reproduction. Ova are haploid, sperm are haploid. So the animal body is made of diploid cells with 2 copies of each chromosome, and the haploid cells only function in sex. In the primitive plants, it is different. In fact, in mosses, the photosynthetic free-living part is haploid. It's as if sperm or ova, let's say, grew up into separate individuals, multicellular, and then they produced the testes and ovaries, which would allow for the next generation of diploid cells. In ferns, the prothallus is the gametophyte stage. These cells are all haploid. Once again, odd for animals because we think of haploid cells only in terms of sperm or ova, but in ferns it is a free-living, typically photosynthetic individual. The prothallus, this gametophyte, then produces the archegonia, where the eggs are made, and the antheridia, where the sperm are made. And the sperm have flagella and, using water, swim to fertilize the eggs. Once the eggs are fertilized, there is now a diploid cell that will develop into the diploid sporophyte phase of the generation, what we recognize as the fern. In the sporangia, meiosis will occur where the haploid spores are produced, which will then start the cycle all over again. In the land plants, we see a gradual transition starting in mosses and liverworts, where the haploid gametophyte is dominant and the sporophyte depends on it. And as we progress through the plant groups, by the time we get to seed plants and the flowers especially, it has been reversed, where the diploid sporophyte is dominant and the haploid gametophyte depends on it and is only used for sex, as in the higher animals. However, one could argue that the gametophyte still serves a function. So because there is only one copy of all genes, then the genes which are expressed would then cause mutations to be selected against. So if a fern gametophyte expresses a mutation that has a harmful effect, it is less likely to reproduce, and thus the mutations can be eliminated from the population more quickly.